I'm Jane Morris, and in this presentation I'll explain what we mean by RNA interference or RNAi, how it works in genetic engineering, and we'll look at possible risks associated with the technique. So, in this presentation we'll cover, firstly, what is RNAi and how does it work? What is it being used for? What are the risks associated with the technique? What has received regulatory approval to date? And I'll then give an example of the risk assessment for a potato engineered with RNAi. So let's start with a look at what is RNAi. It's a mechanism that is used to inhibit gene expression by neutralizing targeted messenger RNA molecules. It can involve two different types of RNA, microRNA and small interfering RNA. An important point is that it's a natural mechanism that plants use to control gene expression during development, in response to stress factors, and to protect against infection. But scientists have now harnessed this natural mechanism to modify a plant's properties. In fact, conventional plant breeders have unknowingly been using RNAi for years, but now it can be used in precise ways through genetic modification. Let's look first at how naturally occurring microRNA and small interfering RNA work. We'll start with microRNA as shown in this diagram. Naturally occurring microRNAs derive from primary transcripts produced by RNA polymerase II, containing imperfect foldbacks, shown here as primary microRNA. Primary microRNAs are cleaved by an enzyme known as a dicer-like enzyme, or just dicer for short, shown here as DCL, in a two-step process producing in succession a microRNA hairpin precursor and a mature microRNA duplex. MicroRNAs act as a specificity determinant of a protein RNA-induced silencing complex, shown here as RISC or RISC. One strand of the duplex RNA is incorporated in the RISC complex. This strand guides RISC to target messenger RNA. The catalytic subunit in the RISC complex, called Argonaut, is responsible for the guided cleavage of the target transcript between positions 10 and 11 of the microRNA. The cleavage is followed by the complete degradation of the sliced transcript and so results in downregulation of the gene. Unlike microRNA, the precursor of short interfering RNA in the plant is a perfectly complementary hairpin-shaped double-stranded RNA. Like the microRNA, it's cleaved by dicer to form short interfering RNAs of 24 to 26 nucleotides. And again, like microRNA, one strand is incorporated into the RISC complex to guide RNA cleavage. Unlike microRNA, however, plant short interfering RNAs of 24 to 26 nucleotides additionally function as a sequence-specific guide in transcriptional gene silencing, consisting of downregulation of transcription by methylation of DNA homologous sequences. A very efficient approach in the lab to inducing gene silencing with short interfering RNA is the utilization of hairpin constructs expressing double-stranded RNAs homologous to target transcripts. Two DNA sequences are placed in an inverted orientation with a short spacer in between. These are homologous to the messenger RNA target. They're transcribed to form double-stranded RNA, which is processed by dicer to produce a randomly assorted population of short interfering RNAs. The length of the inserted sequences that constitute the arms of the hairpin construct can be as short as 40 nucleotides. Hairpin constructs represent a valuable tool for gene silencing because hairpin construct-induced downregulation of target genes is very efficient, occurring in 70 to 100% of independent transformed lines. In addition, the silencing effect is stably inherited from one generation to the next. Scientists also use artificial microRNAs as another means of gene silencing. The microRNA expression cassette consists of the structural backbone of a natural microRNA precursor, where the stem region is replaced with microRNA sequences designed to target a specific gene. It works exactly the same as naturally occurring microRNA and results in the degradation of the messenger RNA target. It can be used to silence any gene in the plant. One advantage of GM crops produced using RNAi is that they can be classed as intragenic rather than transgenic. By intragenesis, we mean transformation with a gene construct made with sequences derived from the same species or from a sexually compatible species. However, the intragene is not a perfect copy of a natural gene. This differs from cisgenesis, where a complete natural gene is introduced. In both cases, there is usually no foreign DNA, unlike in transgenic plants, so they may be more acceptable to public opinion. There are many potential applications of RNAi, and no doubt more will emerge. Plants can be engineered to silence genes that are essential to crop pests and diseases, providing the plants with resistance. And an important application is to modify metabolism to improve nutritional value or to make plants more useful for industrial applications. In addition, RNAi-based insecticidal sprays are in development that would not involve direct modification of the plant. So, are there any risks that we need to be concerned about? Well, one major thing to look out for is off-target effects, though this is less of an issue with microRNA than with short interfering RNA. One problem is that RNAi can potentially silence the correct gene in the wrong organism. So, for instance, this would be a problem if RNAi targeted against an insect pest silenced a gene in a non-target or beneficial insect. For this reason, where RNAi is to be used as an insecticide, knowledge of possible target sequences in non-target insects is important. Another possible unintended effect is the spread and amplification of the RNAi, since in the process of RNA degradation, secondary short RNAs are generated. Although microRNAs generally don't move between cells or tissues, short interfering RNAs have been shown to spread systemically in plants. And so even where tissue-specific expression is the goal, possible silencing effects throughout the plant should be considered. The risk assessment for food and feed for an RNAi-modified plant has to be handled somewhat differently from a transgenic plant. Whereas the GMO risk assessment usually focuses on the safety of newly expressed proteins, RNAi results in decreased expression of endogenous proteins and no new proteins. For this reason, toxicity and allergenicity studies are not considered particularly valuable. In addition, if nutrient profiles and composition are comparable to conventional varieties and there is no change in bioavailability, then animal feeding studies are generally not considered necessary. In addition, RNAi has a history of safe use. Many species in nature commonly use RNAi to regulate gene expression, and this trait is highly conserved in many species. Many of the traits that have been produced through conventional breeding in fact use endogenous RNAi. Examples include soybean seed color and rice protein content. So bearing this in mind, the regulatory consensus is that consumption of RNA including double-stranded RNA and short interfering RNA is safe, since we have been eating the naturally abundant forms for many years. In fact, Food Standards Australia-New Zealand stated that there is no scientific basis for suggesting that small double-stranded RNAs present in some GM foods have different properties or pose a greater risk than those already naturally abundant in conventional foods. Bearing all these facts in mind, we could consider that much less stringent regulatory oversight would be required for RNA-modified crops than for transgenic crops. RNAi-modified crops are only now coming to the fore, but there are many of them in the pipeline. Those with regulatory approval in various countries, as shown here, include potatoes, apples, lucerne, beans, papaya, pineapple, and maize. I'll cover the risk assessment for the innate potato, the first one on this list, as a case study for an RNAi-modified crop. This slide shows how the innate potato has considerably reduced browning and discoloration after peeling, but it also, in addition, has reduced acrylamide potential. Let's start by looking at the reduced acrylamide potential. Acrylamide is a carcinogen that is formed from a reaction between the amino acid asparagine and reducing sugars when potatoes are cooked at a high temperature, such as baking or frying. It's not present in fresh potatoes. To reduce the problem of acrylamide synthesis, the innate potato contains inverted repeat segments of 3 genes involved in acrylamide biosynthesis. One segment was derived from a gene that codes for an enzyme catalyzing asparagine biosynthesis. 2 other segments were partial promoter sequences of 2 genes encoding proteins involved in converting phosphorylated starch to reducing sugars. Then, in addition, to reduce the browning problem, the innate potato contains inverted repeat segments of a gene coding for polyphenol oxidase. So, what was considered in the food safety assessment? Well, first of all, bioinformatics analysis looked at the junction sequences of the insertion to check that there were no open reading frames that could encode any polypeptide showing homology to known allergens or toxins. A single contiguous amino acid sequence was identified, identical to a sequence from a known allergen from avocado, but this was identified as a minimal hazard. Then, potato tuber composition was analyzed from multiple locations in the United States during the years 2009 to 2011. Tubers of the GM potato varieties, controls, and reference varieties were planted in a randomized complete block design consisting of up to 5 blocks per site. A total of 52 components were analyzed according to the OECD recommendations for potato. The components included proximates, amino acids, vitamins, minerals, glycoalkaloids, sucrose, and reducing sugars. Statistical differences were found, but they were within the previously determined tolerance levels and published ranges in the literature. The intended reduction in levels of free asparagine and reducing sugars was found near the time of harvest in most events, but the differences in reducing sugars were not sustained after 2 to 5 months storage. However, it was significant that the levels of acrylamide in cooked potatoes were reduced by as much as 60% compared with controls and conventional potatoes. The conclusion then was that no safety or nutritional concerns were raised, and that the GM potato was as safe and nutritious as the conventional counterpart. Then, the environmental safety was also considered. Firstly, no differences in agronomic characteristics were noted in comparison with the conventional counterpart. There was also no indication of altered weediness potential. The russet Burbank potato variety that was used is sterile, so there can be no gene transfer. However, in the case of other potato varieties, pollen transfer is limited to around 20 meters, and potatoes are generally clonally propagated. Any gene flow to wild relatives would not increase their weediness. So we reach the overall conclusion that the innate potato is safe for environmental release and consumption. So, I finish with a few final thoughts about RNAi. First, we should note that crops modified with RNAi are relatively new to the market, and they aren't yet available in southern Africa. However, this is set to change because there are enormous opportunities and many new crops are in the development stages. In general, regulatory requirements may be less stringent than for transgenic crops due to the lack of newly expressed proteins. It's also worth noting the detection of our RNAi modified crops is only possible if information about the inserted sequence is provided by the producer. So there's no generic methodology to test for the presence of unapproved events. When regulators in the Southern African region are considering how to develop or improve their regulations, they need to make provision for crops that don't produce new proteins and where the risk is generally low. The only real concern is with regard to the use of RNAi for control of insect pests, where more investigation is needed in terms of possible effects on non-target organisms. I hope you found this overview useful. Thank you for watching.